talking about translation today, and it's a pretty dense topic. And so what we're going to do is we're going to break this part of the chapter up into two sections. So the 17.4 is going to have two separate videos. The first video is going to introduce the tRNA and the ribosomes, so just sort of the key players. And then the second video is going to go through initiation, elongation, and termination. So let's get started. The kind of unsung hero of tRNA is the anticodon. So we talk about the ribosome being the place where protein synthesis happens. But really what's doing the actual translating in this process is the tRNA. And so if we look at the molecule here, we'll see that there is an attachment site for the amino acid, and the anticodon is at the bottom. And this is the actual translation happening here. This is what's translating between the language of nucleotides to the language of amino acids. It's this right here. And so let's take a closer look at the amino acids. The tRNAs. The tRNAs are a single strand of nucleotides, about 80 nucleotides long. And if we look at it in a 2D shape, so it's unfolded, and you can see that it looks like this clover sort of, and it's got 3 loops, and at the bottom loop is our anticodon. And then we have the 3' and the 5' end that are lying next to each other, and the attachment site is on the 3' end. When we let the hydrogen bonds between the base pairs align and bond, we'll see that it takes on this 3D shape, once again similar to protein folding. There are these intermolecular forces are going to cause the 3D shape to form. And this is allowed by the mRNA because of these long segments of base pairs, or open base pairs, because it's a single-stranded molecule. When we're looking at tRNA in the book, we're going to see that it looks like this. Here's the attachment site for the amino acid, and here is our anticodon. Anticodons are written in a 3' to 5' direction to align with the codons that are written in a 5' to 3' direction. So let's take a step back and let's think about this. DNA, the template strand, is written in a 3' to 5' direction. When we replicate DNA, we are always adding on to the 3' end. So we're going to be always adding on to the 3' end. So replication happens in a 5' to 3' direction. That means that the template strand is written in a 3' to 5' direction. And so that means when we make RNA or an mRNA strand, the antiparallel, or the complement to the 3' to 5', right here is going to be 5' to 3'. And so when we find the complement to that, the anticodon is going to be 3' to 5'. The codon on the mRNA, so right here, our codon on the mRNA is going to be matched up with a specific anticodon. We can see this happening here. Here's our mRNA strand, and here's our anticodons, which are lining up with a specific codon. The tRNAs are going to line up with the codons, or the anticodons are going to line up with the codons. And they're going to bring this amino acid, drop it off, and then go back into the cytosol. And so now multiple tRNAs are going to try and line up here at this very specific codon. But because it doesn't match, they're going to try and fit, and then they'll go away. And another one will try and fit, and then they'll go away, until the right tRNA comes along and then it fits, which actually happens at an amazing speed, considering all of these steps. So we are looking at tRNA, and let's look at the 2 points in the translation process where molecular recognition happens, where we have to recognize tRNA in another molecule to bind it. And the first part is in the cytosol. the tRNA, here's our tRNA molecule, our little L-shaped molecule, is going to be bound to an amino acid. It needs to be covalently bound to a very specific amino acid. And there's going to be a specific enzyme from a family of enzymes called the aminoacyl tRNA synthetases. And so this should make sense. Aminoacyl or amino acid, tRNA, building of. So we're going to build these. It takes one ATP to bond an amino acid to a tRNA, which we call a charged tRNA. And so there are 20 different enzymes which go with the 20 different amino acids. However, there are 64 possible codons. 61 of those are going to be paired with an amino acid. And so we would think that there should be 61 enzymes, but there's not 61 enzymes. There's 20 enzymes. And so that means that multiple codons are going to be able to be matched with amino acids in the enzyme. So let me say that differently, that this enzyme is going to be able to read and pair GCC with alanine. It's also going to be able to read and pair GCG with alanine. So there's one enzyme per amino acid. And so it's going to be able to read multiple codons for the, or anticodons for the binding of the two. All right. The next place where molecular recognition is going to happen is in the actual process of translation when the anticodon right here is matched with the codon. So our molecular recognition. And so here we have our tRNA, our anticodon, and you can see here's our first base pair, our second base pair, and there's something funny with our third base pair. The first two nucleotides are going to line up, and there's not a lot of flexibility here. The A has to match with a U, the G has to match with a C. However, this third base pair is going to have what we call the wobble effect. It's going to be a little bit more flexibility. It's going to be a little bit more loosely aligned. And so therefore, they're going to be able to swap out, not really swap out, the wrong base pair sometimes is going to line up and it's going to be acceptable and it's going to pass through. And so if there were a single tRNA, so a single tRNA, for each mRNA codon, we would expect that there would be 61 tRNAs for each codon, right? So we said that there's 61 codons for our 20 amino acids. And so if there's 61 codons, we'd think that there's going to be 61 tRNAs. However, there are, in some bacteria, there are only 45 different tRNAs. And that means that sometimes this wobble effect is in place, that there aren't actually enough tRNAs to read the actual number of codons on mRNA. And so this is one way that is a fix for this problem. And so we call this flexibility a wobble, the wobble effect. And if you want to learn a little bit more about that, there's a video here, and I'll post that on PowerSchool for you. So we've talked about tRNAs. Let's talk about ribosomes. Ribosomes are composed of a large subunit, and a small subunit. They're also made of proteins and rRNA, ribosomal RNA. And we talked about how rRNA, or when RNA acts as an enzyme, when it's a catalyst to a process, to a reaction, then we call it a ribosome. And so a ribosome is actually a colossal ribosome. So it has proteins and rRNA. And there are thousands of these ribosomes in a cell, and therefore rRNA is going to be the most abundant type of RNA in a cell, because there is a lot of RNA per ribosome. Only a third of the mass of a protein of a ribosome is made of proteins. The rest is rRNA. In a bacterial molecule, 3 RNA molecules exist, and in a eukaryotic ribosome, 4 rRNA molecules exist. And so here we can see our large subunit and our small subunit paired together. And there's going to be a very specific structure of the ribosome because it has a very specific function. And so right here, this is where the mRNA is going to bind. We can see the mRNA binding site is here. And then this is where our tRNAs are going to bind. There's the A, the P, and the E site. So tRNAs are going to enter in, and like we said, it's going to go into the A site, and it's going to try and match the anticodon with the codon. If it's a fit, it's going to be translocated to the P site. The P site is where the amino acid is added to the growing strand. It's going to be added to the carboxyl end of the amino acid. What does this mean? Here's our N-terminus, or our nitrogen, and here's our carboxyl end, our C-terminus. And so when the amino acids are added to the growing strand of amino acids, or to the polypeptide strand, they're going to be added to this side, to this side, to this side. And so that's going to happen here in the P site. Then, once that has been added, once the amino is added to the growing strand of the polypeptide, then it's going to translocate to the E site, which E stands for exit. Once it goes into the E site, it floats off. It goes back into the cytosol. It goes back to the aminoacyl tRNA synthetase and is going to be recharged. And so here we are at initiation. So I'm going to stop that video and we'll start a new one with this process.